Hi everyone, welcome back to my A-Level Biology lecture series. In this lesson, we will explore light-independent reactions. At the end of this lesson, you will be able to outline the three main stages of the Calvin cycle. In the process, Rubisco catalyzes the fixation of carbon dioxide by combination with a molecule of ribulose bisphosphate, RUBP, a 5-carbon compound, to yield 2 molecules of glycerate 3-phosphate, GP, which is a 3-carbon compound. GP is then reduced to triose phosphate, TP, in reaction involving reduced NADP and ATP. In the next step, RUBP is regenerated from TP in reactions that use ATP. And state that Calvin cycle intermediates are used to produce other molecules, such as GP to produce amino acids and TP to produce carbohydrates, lipids, and amino acid. Let's get started. Light-independent stage of photosynthesis is known as Calvin cycle. It is a set of anabolic reactions that occur in the stroma of chloroplast and do not require light energy directly. It is comparable to the Krebs cycle as it is a cyclic pathway. So basically, the starting material or starting compound will be regenerated back at the end of the reaction. In this case, in Calvin cycle, initial carbon acceptor is regenerated. However, the Krebs cycle is a catabolic reaction, oxidizing acetyl coenzyme A and produce ATP. However, the Calvin cycle is anabolic, where ATP is used to build triose phosphate from carbon dioxide. This reaction use ATP and reduce NADP, and these two compounds are produced during light-dependent reaction to fix carbon dioxide into organic compounds such as triose phosphate. Generally, the Calvin cycle involves 4 key stages. The first 3 stages are involved in the Calvin cycle. However, the last phase actually is part. It's not really a part of cycle, but it is a branch from the cycle. So the first process is known as carbon fixation, followed by reduction and regeneration of RUBP. These 3 stages are part of the Calvin cycle. The process of product synthesis branch out from this Calvin cycle. Let us look at the reaction. During carbon dioxide fixations in Calvin cycle, ribulose bisphosphate, RUBP, with 5 carbon reacts with carbon dioxide to form an unstable 6-carbon intermediate compound. This reaction is catalyzed by an enzyme known as Rubisco. This unstable 6-carbon intermediate compound immediately dissociates to form 2 molecules of glycerate 3-phosphate, which is also known as GP. Each molecule of glycerate 3-phosphate has 3 carbon atoms. In the next phase, which is reduction phase, glycerate 3-phosphate GP, in the presence of ATP and reduced NADP, glycerate 3-phosphate is reduced to form triose phosphate. In this phase, ATP is hydrolyzed to form ADP, and reduced NADP is oxidized so that NADP can be regenerated. Some of these triose phosphates are used to regenerate back ribulose bisphosphate or RUBP. In these reactions, ATP is needed. Therefore, ATP is hydrolyzed to form ADP. Some of these triphosphate is used to form glucose molecule with 6 carbon. In order to form one glucose molecule, Calvin cycle has to take place 6 times. Therefore, in this diagram, you can see that overall reactions require 6 ribulose bisphosphate to react with 6 molecules of carbon dioxide to get 6 molecules of unstable 6-carbon intermediate compound. And these 6 unstable 6-carbon intermediate compound dissociates to form 12 molecule of glycerate 3-phosphate, and 12 molecule of glycerate 3-phosphate GP are reduced to form triose phosphate. A total of 12 triophosphates are formed in 6 cycles of Calvin cycle. Out of these 12 molecules of triose phosphate, 10 molecules of triose phosphate with a total of 30 carbon are reorganized and to regenerate back 6 molecules of ribulose bisphosphate, and the remaining 2 molecules of triose phosphate are used to form products in the process of product synthesis phase. So again, let us go through the process. Phase 1, carbon dioxide fixation or carbon fixation, where each carbon dioxide molecule is attached to a 5-carbon compound known as ribulose bisphosphate or RUBP. So this reaction is catalyzed by the enzyme, a very important enzyme, Rubisco. An unstable 6-carbon intermediate compound is formed, and this unstable intermediate compound immediately splits or dissociates into 2 molecules of glycerate 3-phosphate. It is very important to mention for every one unstable 6-carbon intermediate compound, 2 molecules of glycerate 3-phosphate are formed. In phase 2, which is the reduction phase, each GP or glycerate 3-phosphate molecule is phosphorylated by ATP and then reduced by reduced NADP to form triose phosphate. For every 6 carbon dioxide molecule, 12 TP molecules are formed. Next phase, which is termed as regenerations of RUBP, where out of 12 molecules of TP, 10 molecules of TP are used to regenerate 6 molecules of RUBP. This step consumes 6 ATP molecules. RUBP is then ready to accept more CO2, allowing the cycle to continue. Phase 4, which is product synthesis phase, where every 2 TP molecule form as a net yield, the Calvin cycle consumes 18 ATP and 12 reduced NADP. The TP produced is used to synthesize glucose, sucrose, and other organic compounds through additional pathways. Next, let us look at the utilization of triose phosphate in plants and how plants actually utilize triose phosphate to synthesize organic carbon compounds. First of all, let us look at starch synthesis and storage. Some TP is used to synthesize glucose, as we have learned previously, and this glucose is polymerized to form starch, and it is stored inside the chloroplast. Inside the chloroplast, this starch is accumulated and forms starch granule. When required, starch is hydrolyzed and forms maltose, and the maltose is transported to the cytoplasm and cleaved into the glucose 2 or 4 aerobic respiration to take place. And next, let us look at biosynthesis of essential molecule. TP molecules are diverted into pathway to synthesize, for example, amino acid to form protein, fatty acid and glycerol to form lipids, nucleotides to form DNA and RNA, as well as some other chemicals such as pigment, for example, chlorophyll and carotenoids for photosynthesis. And these biosynthetic products are essential for self-growth, metabolism, as well as photosynthetic function. Sucrose synthesis and transport. Some TP is transported to the cytoplasm where it is converted into sucrose, where this TP, 2-TP forms glucose, and glucose combined with fructose to form sucrose. And sucrose is actively loaded into the phloem and moved by translocation to other parts of the plant. So in the recipient cells, also known as at the sink, sucrose is hydrolyzed into glucose and fructose, which this glucose and fructose can be used in aerobic respiration. However, before fructose can be used, fructose must be converted into glucose. And the glucose can serve as precursor, basically means the starting material for amino acid, lipids, and carbohydrates. TP produced in the Calvin cycle is used to synthesize glucose, which exists in two isomeric form. As we have known, glucose can be in alpha glucose or beta glucose form. So this glucose monomer form storage as well as the structural polysaccharides in plant. Storage carbohydrates include starch. So starch is the polysaccharide formed by the condensation of alpha glucose molecule, and it serves as the primary carbohydrate storage in the plant. So the starch can be stored in the root or some storage organs such as the seed as well as the tuber, for example, potato tubers. How about structural carbohydrate? Yes, structural carbohydrate actually is cellulose. So cellulose formed by the condensations of beta-glucose molecule, and cellulose is found in the plant cell wall, which provide rigidity and support to plant cells to resist the excess entry of water molecule in order to prevent the bursting of the plant cells. With that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any questions, feel free to drop them in the comment section. See you in the next lesson and have a great day.